Hello. So it will not let me download my video for some reason, so I'm just gonna try to record my slideshow and do my notes. This is not ideal, but we'll see how this goes. Okay. Hello and welcome. Today we are diving into DNA barcoding, a groundbreaking molecular technique that's transforming the way scientists identify and classify species across ecology, forensic science, and many other fields. For centuries, species identification was based mainly on morphology, the physical characteristics of organisms, such as shape, size, or color patterns. While this method remains valuable, it has clear limitations. Some species, called cryptic species, look nearly identical, but are genetically distinct. Juvenile forms often differ significantly from adults, confusing identification. Additionally, morphological analysis requires expert taxonomists and can be slow, subjective, and prone to error. DNA barcoding addresses these challenges by using a short, standardized region of DNA to identify species accurately and efficiently. Imagine a supermarket barcode. It uniquely identifies a product by sequence of numbers. Similarly, DNA barcoding uses a unique genetic barcode encoded in a specific gene segment to pinpoint an organism's species identity. The success of DNA barcoding hinges on several scientific principles. The barcode region must be universally present in the target group of organisms. It needs to show enough genetic variation between species to allow discrimination, and at the same time, it must have low variation within a species so individuals of the same species share similar barcodes. For animals, the mitochondrial gene cytochrome C oxidase 1 or CO1 is the standard barcode. Mitochondrial DNA is ideal because it is abundant in cells, inherently maternal without recombination, and evolves at a rate that balances conservation and divergence, fast enough to distinguish species but stable enough within them. In plants, researchers use regions of the chloroplast genome such as RBCL and MATK because plant mitochondrial DNA evolves too slowly for effective barcoding. Now, let's explore the process of DNA barcoding step by step. Sample collection. First, a small piece of tissue is collected. This might be a leaf, a piece of fish muscle, or even trace DNA left in water or soil. Number 2, DNA extraction. The genetic material is carefully extracted using chemical and mechanical methods to obtain pure DNA. PCR amplification. Next, the barcode gene region is amplified using PCR. This technique cycles through temperature that separates DNA strands, attacks primers specific to the barcode region, and replicates the DNA millions of times. Number 4, sequencing. The amplified DNA is then sequenced, meaning scientists determine the exact order of the nucleotides, the A's, the T's, the C's, and the G's that make up the barcode. Number 5, database comparison. This sequence is compared against reference databases like the Barcode Light of Life Data Science System, BOLD, or GenBank, which contains millions of barcode sequences from known species. Number 6, species identification. Based on the best match, the specimen species is identified with high confidence. While DNA barcoding is revolutionary, it has limitations. The technique depends on comprehensive and accurate reference databases. If a species is represented or data is incorrect, identification can fail or be ambiguous. Some closely related species share very similar barcode sequences, complicating differentiation. Moreover, the decreasing use of DNA barcoding in forensic and commercial contexts raises ethical questions about privacy, data ownership, and consent, especially as genetic data becomes more widely accessible. Responsible use requires clear guidelines and regulations. The future of DNA barcoding is exciting. Scientists are now integrating it with environmental DNA, or eDNA, analysis, detecting DNA fragments organisms shed into water, soil, or air, enabling non-invasive biodiversity assessments without physically capturing organisms. Advancements in next-generation sequencing allows simultaneous analysis of thousands of species in complex environmental samples, known as metabarcoding. Portable sequences sequencing devices are making barcoding accessible outside traditional labs, opening new frontiers for rapid field identification. Additionally, artificial intelligence is being applied to analyze large barcode datasets, improving species discovery and monitoring in real time. In conclusion, DNA barcoding is a powerful molecular tool that decodes the genetic signatures of life, enabling precise species identification that underpins conservation, food safety, forensics, and ecological research. As technology advances, its impact only will grow, deepening our understanding and stewardship of the natural world. Thanks for watching. I apologize for the shaky handwork. I tried my best, but this is definitely not ideal. I apologize. It just will not let me download for some reason, which is quite annoying. But yeah, okay, hopefully this suffices.